Hello. Hello. Can you hear me? Hello. I don't know if you guys can hear me. Once you guys can hear me, I'll be able to start. So good morning, everybody. I work at Memorial Sloan Kettering Cancer Center in New York City. Today, I'm going to talk to you about integrative um, and integrating innovative biobanking and learning from human biobanking to inform animal and environmental One Health. So I'll be starting actually with the human biobanking, and let's see how far we can go, because I have only 20 minutes and there is a lot to cover. And so I'm unable to move my slide forward. So now this slide represents major landmarks in genetics and genomics. But the most important that I would highlight today is the one in 1990. So in 1990, the Human Genome Project was launched in the United States, and that started the entire process, which was actually uh, went went on for about 13 years before the human genome sequence was actually completed. And it was along the way there are many landmarks. For example, the establishment of Human Genome Research Institute, and then the full-scale human genome sequencing project began in 1999, and the first draft of the Human Genome Project was published in 2001 in two journals, that is the Science and Nature. So this was a historic moment when the Human Genome Project was completed, and actually it changed the way science and medicine works today. So the Human Genome Project first draft, now it has been about quarter of a century ago that this was done. So this project has empowered research into the molecular genetic basis of human diseases, and it has completely revolutionized the drug discovery pipeline, and also it has helped to revise the idea of the gene itself. Now this is the MIT Center for Genome Research, where you can see this looks like a assembly line in a factory where the genome sequencing was actually done. Dr. Eric Lander, who was the director of Whitehead Institute at that time, said, generating the complete sequence of human DNA is the most exciting adventure in modern science. And I think many of us will agree with that. Now, Human Genome Project took about 13 years to complete, and it cost $3 billion. Now, the cost of human genome was expected to go down according to the Moore's law here. So, but unexpectedly, it actually dropped pretty significantly. You can see here the cost went down, and now the cost of human genome is under $500, and it takes about a week to complete. Now, 20 years after the Human Genome Project was completed, it was a time when, Bates et al., Gates et al., they decided to evaluate where we stand today. So 20 years later, it was realized that this Human Genome Project has a deep impact. So now, in this particular diagram, you can see the year 2001, which is represented by a plane. So if you look at the events on above the plane, those are the events before 2001, and those beneath the plane are the events that happened after the Human Genome Project was completed. So in terms of the number of publications and which genes investigators focused on, this shows that most of the research was focused on the top 8 genes, which are represented on the underside of the plane. So there are about 19,757 protein coding genes, and at that time it was thought that the non-coding elements, which are called junk DNA, would not have any role to play in the biology, but it proved otherwise. Actually, the non-coding elements help to regulate the dynamic code of life. So after 2001, the researchers had a lot of interest in exploring the non-coding elements. So therefore, there was an explosion of interest in non-coding regions of the genome after 2001, which is shown in this graph, where you can see there are about 130,000 publications on these non-coding regions compared to only 20,000 of the protein coding regions of the DNA. So, like I already said, there were about 8 genes which became the star genes, and the topmost was the P53, followed by EGFR, TNF, interleukin 6, VEGF-A, CD4, HbA1, and others. So these top 8 genes became the center for every research, and partly it was due to the researchers being able to secure the funding. So Human Genome Project was expected to expand genetic discovery, but then the research mainly focused on these 8 genes. So after that, it also came out that around the year 2000, only 50% of the drugs actually knew their target. So whatever drugs were used had a specific target in only 50% of the cases, but after 2001, nearly 100% of US drugs licensed in any given year had all the potential proteins target identified. So you can see here, all of the drug targets were known after 2001. So this is the tremendous impact Human Genome Project had on molecular research. Now, Human Genome Project, according to me, also changed the drug discovery and clinical development pipeline. So drug discovery is a very long process. So it's a very lengthy, risky, and expensive. So partly because only 1 out of 10 drug usually succeed to reach approval. So and partly the reason for that is gaps in our understanding of the tumor biology and of in general of human disease, and then excessive reliance on non-human models, and prioritization of targets and agents with low specificity. And partly also pharmacology plays a role, and also sometimes their toxicity is not very well understood. So that's why many of the drugs fail to to get approval. So that makes the drug discovery process very long and expensive. And it was hoped that after identifying the targets for therapy, the drug discovery pipeline might get accelerated and also become less expensive. So now I'm moving to MSK Biobank, where I work. So MSK Biobank, I'm going to give you a quick overview. So this biobank works uh, like this, starting with patient consent. So consenting patients is done at the initial point of contact, and then specimens are collected, but our collections are DMT driven, which means disease management team driven. So it is thought that this is a better way of collecting tissues, and which centralizes the clinical and translational samples. And because the hospital has many different locations, so transportation plays a key role. So there is a unifying transport teams under the biobank which transport the samples to the central biobank. So once the specimen reach the central biobank, the clinical and research data are integrated, and after that the specimens are processed using our standard SOPs, and then they are banked, and then the samples are distributed to researchers who have been approved and those who have obtained approval for their usage. And the biobank also plays a part in education. So MSK Biobank long time ago adopted these interactive dashboards. So these dashboards are available to investigators and who can actually access the information about the samples they are interested in using these dashboards. So as you can see here, melanoma, we had a melanoma biobank. Then there was a hematology and hematologic and oncology tissue bank, and PPBC. Now we have integrated all these three as a single central MSK biobank. Now this is the dashboard which is used by the people who work in the biobank. So this allows us to uh, locate the samples, also look up the protocols, which protocol and consent this particular sample is assigned to. And also we can look at specimen units, specimen types, histological diagnosis, and also position, shelf, location. And also we can also look up the images, where in some of the samples we also have histological images associated with those samples. So all these can be accessed using these dashboards. Now MSK Biobank also supports an experimental immuno-oncology biobank, which is where we actually dissociate tumor tissues, and we convert the tissues into single cell suspensions. So these single cell suspensions are aliquoted and they are stored, and they are made available to MSK researchers who are working in the area of immuno-oncology. So far we have collected about somewhere around 4,000 samples from about 3,500 patients, and the project actually started in April of 2021. So it has a slow start, but still it's gaining ground. Now this is one more thing I wanted to emphasize, that the way the tumors are acquired by the biobanks has changed drastically over the last few years. Now, traditionally, the pathologist and surgeon used to play a part in the biobanking, but now It is the interventional radiologists, surgeons, pulmonologists, dermatologists, and other physicians who play increasingly they play a role in biobanking. And because of this reason, what happens is we are able to acquire very targeted samples which are radiologically guided. But these are very small samples. These are core needle biopsies. So the sample size has decreased significantly. And in the traditional biobanking, it used to be the primary disease, but now it's the primary. There is a recurrent disease and metastatic disease. So sometimes these are collected only in the form of biopsies because the patient is already on therapy. So the amount of sample used to be ample, but now the samples are scant. They are small. And there is a increasingly more demand on these samples because these samples are needed not only for histology, but also for genomics. They are needed for immunohistochemistry and also for industrial collaborations, for clinical trials. So this particular scenario makes it difficult to do too many things with these small samples. So earlier, the sample used to be collected at the initial time of diagnosis, but now the samples are collected at multiple time points during the disease process and also the treatment trajectory of the patient. So there are multiple samples collected, like tissues, there is blood, there is urine, collected at different time points during the course of the disease. Now, samples are also collected in different ways, like FFPE, flash frozen. They are collected in special preservatives and embedded in OCT. Now, the pre-analytical variables also depend on what the specimen is going to be used for, and sample sharing has become infrequent because the samples are too small. But the positive thing is all these samples have very detailed clinical information associated with them. Now, like I said in the beginning, there was a whole genome sequencing approach, and then it took several weeks to sequence the entire genome. But now slowly the sequencing can be completed within few days. So in our group, we decided to experiment whether whole genome and transcriptome profiling can be used for clinical oncology as a clinical tool. So it was observed that the entire process could be completed within 9 days. So increasingly, we are coming closer to the point when whole genome and transcriptome profiling can be used as a standard of care in the United States. So we are very close to those days when whole genome and transcriptome can be profiled in less than a week. Now, certain diseases like small cell lung cancer, where the tissue is limited, and when the disease becomes metastatic, it's very difficult to obtain those samples because they are not resected. So in those cases, patient-derived xenografts and circulating tumor cell-derived xenografts are tried, and we found that these were pretty successful, and they are very good representative of the original disease. Now, biobanks are nothing but these are freezers full of hope for humanity. So this article appeared in the Times in England, and this actually pertains to the UK Biobank. So population-based biobanks it's expected that they will be playing a major part in precision medicine. So personalized therapies rely on the data with substantial statistical power to map the genetic and epidemiological associations that create models to inform clinical decisions. So biobanks have emerged as large-scale longitudinal cohort studies with long-term storage of biological specimen and very rich health information, including electronic medical records and participant surveys. So biobanks are in a position to support ongoing and precision medicine initiatives by genotype phenotype mapping efforts, pharmacogenomic studies, polygenic risk score assessment, and rare variant analysis. Now, this is a diagram representing what I said already, but here I want to emphasize that biobanks are also in a position to play a part in emerging public health needs. So this is possible only when we have collected samples from the population, which represent the population at different time points. So if the samples are collected in large numbers and are good representative of the population, then this specimen or the biobank can be used for a precision medicine for all of society. Now, this is the global distribution of population-based biobanks all over the world, and here the only biobanks which share their data freely with investigators all over the world, they are represented here. So some of these biobanks are in the United States, you can see. Then there are some in England, United Kingdom, then Finland, Estonia, Qatar Biobank, China, Kaduri Biobank, and the Biobank Japan. So only these biobanks are shown because they freely allow researchers to access their data. And these are public biobanks. And all of them are very large biobanks. Minimum collection is about 50,000 samples. So these are representative of the population of that particular country or region. Now I'll be showing you more about so these biobanks collect blood, saliva, pathology blocks, stool, urine, CSF, breast milk, dental plaque, nail fragments, and disease tissues. And in addition to that, they also include participant surveys on lifestyle, mental health, substance abuse, substance use, and dietary factors. So these participant surveys happen at several occasions. It's not just one survey. They occur at different time points. Now the types of data in the UK Biobank. So UK Biobank is started with about 500,000 men and women aged between 40 to 69 years, and they were recruited between 2006 and 2010. So the idea is to study the genetic, lifestyle, and environmental determinants of wide range of diseases like the cardiac disease, obesity, hypertension, and also cancer. So UK Biobank was established by the MRC UK, that is the Medical Research Council, Wellcome Trust, British Heart Foundation, and Cancer Research UK. So now in this diagram, you can see this is the type of information or type of data that this biobank collects, in addition to biospecimen. So it collects data about hearing, vision, brain MRI, cognitive functions, carotid artery ultrasound, MRI of the heart, blood pressure, and collects blood for genetic, that is genotyping, whole genome sequencing, then metabolites, protein, telomere lengths, hematology, biochemical measures, infectious disease markers, whole body bone density scan, and heel bone density ultrasound. So all this, along with the physical activity monitoring, electronic health records. So it's very rich in health information. So in this scenario, it's very important to protect their privacy, the information, the electronic, the personal health, personal information of the patients. Now, the other example of a population based biobank is the All of Us research, in the United States. So all of us, Biobank is an effort to collect data and study data from at least 1 million individuals who are residing in the United States. The project is supported by NIH, and the program aims to accelerate precision medicine by creating one of the world's largest and the most diverse databases of health research. And participants are enrolled at a variety of locations, including mobile sites, hospitals, and walk-in clinics. So once the consented participants are enrolled, so some of them are direct volunteers. They collect the digital health technology, use the digital health technology to collect all the information, and the biological samples are collected, stored in the biobank and electronic health record. And once the biobank sends the samples to genome centers, so all the results of all these assays are returned to the data research center, and along with the associated information of electronic health records and patient information. So all these information is made available to researchers through a researcher workbench. So these researchers can then carry out research based on their subject of interest. So this is how a research is conducted on these samples. So now this is the overview of all of us research project, where there is a return of genomic results to the patients. So participants are enrolled after their consent, and the blood samples are collected, and they are stored at the Mayo Clinic Biobank. DNA extraction is done here. So once the DNA extraction is completed and QC is done, the samples move to the next level, that is the genome center, which is at the Baylor College of Medicine, Broad Institute, and University of Washington. So library construction and whole genome sequencing is done, and after that the data moves to the for variant calling and for pharmacogenomic studies. So after this data analysis, reports are generated which actually show pharmacogenomic report and hereditary disease risk, and the results are returned to the participants who have actually consented for this. So, and they are also provided links for genetic counseling. Now, to make any meaningful information, to get this meaningful information from this data, it's important that the data is huge. Otherwise, we cannot conclude, we cannot make any conclusions about the health of a population if we don't have a large enough data. Therefore, trans-biobank collaborations are very important to advance precision medicine. But then there are considerations like protection of the participants, their information, which need to be considered. But if planned properly, these collaborations can have very fruitful outcomes, like they can lead to novel genotype-phenotype associations, improved polygenic scores, and summary statistics with greater power to advance precision medicine research. Now this is a graph which is quite telling, because this shows the proportion of samples from individuals cumulatively reported by the Genome-Wide Association Studies Catalog in July 2021. So as you can see, on one side you can see the global population, which is in billions, and the proportion of each group. For example, the European population, East Asian. But if you see this particular graph, you can clearly see that the East Asian, South Asian, and African, Hispanic, Latino populations, they form a very small portion of this particular graph. So most of these genome-wide association studies have a very heavy very strong underrepresentation of non-European populations. Now, the lack of genetic diversity in biobanking is something that has caught attention of people around the world, and underrepresentation of individuals of non-European ancestry in biomedical research is a problem, and this results in poor transferability of the results to the population. So any biobanking effort needs to include diverse participants, which will result in a rich genetic data and better precision medicine discoveries. And this is the roadmap. So I think I already have exceeded the time. So I'll, I think, end here. So thank you very much.
डॉक्टर सिंह सुन रहे हैं? डॉक्टर सिंह, हेलो।
I can hear you. Can you hear me?
डॉक्टर सिंह सुन रहे हैं?
Yes, I can hear you very well.
हेलो।
I can hear you. Can you hear me?
डॉक्टर सिंह सुन रहे हैं? हाँ सर, आपकी आवाज।
Yes, I can hear you very well.
हेलो।
I can hear you. Can you hear me?
डॉक्टर सिंह सुन रहे हैं? हाँ सर, आपकी आवाज।
Yes, I can hear you very well.
हेलो।
I can hear you. Can you hear me?
Minimize. Minimize. Cancel. Cancel. Yes, we click on this. Thank you. Yeah. Green. 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 Button.
Maximum.
Can you hear me now? Is my presentation is loaded? Can you hear me clearly? Is my presentation is loaded? Can you hear me clearly? Is my presentation is loaded? Can you hear me clearly? Is my presentation somehow I'm not able to hear back. Can you see me or you cannot see me? यहाँ presentation नहीं जा रहा है ना?
Hello.
Yes, I can hear you.
Sir, there is a button presentation button. I can hear you. Sir, there is a presentation.
उनको बोलिए सर हेलो
Sir,
माइक ऑन नहीं तो माइक बार बार बोलेंगे तो रिपीट
More. Hey, more. Audio. Screen sharing नहीं हो रहा है उसमें.
Hello. Hi, Swati. Somehow I am with it is. Okay.
वो शेयर का नहीं इधर दिख नहीं रहा.
Not a problem. Not a problem. I think ask somebody else to go ahead. Okay. 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 Not a problem.
and it's not significant.
He's a great swimmer. Right? He gave us so many. So this kind of atmosphere, coming together and we are discussing together, always there is a up and downs in time. We should not worry, but we should worry about the notes. And I am confident that our goal will be achieved and people will have to see a good recognition. I congratulate the organizers and all. Thank you very much.
Anita, ये question from
Thank you so much, sir. So now is the most exciting time, that is BBIF Awards 2025. Every year, BBIF committee announces biobanking pioneering awards to individual scientists of exceptional creativity who proposes pioneering and highly innovative approaches with potential to produce an unusually high impact in the field of biobanking in India. So I take the privilege to announce this year BBIF Pioneering Award 2025 that goes to none other but Dr. Anita Mahadevan. A huge round of applause for her. I further take the opportunity to invite Honorable Dr. Anita ma'am to present this prestigious award to all our BBIF awardees. So ma'am, please.
कोई नहीं, उन्हें दिखेगा नहीं। और दूसरी चीज ये है, अगर कोई क्वेश्चन पूछता है ना तो उसमें टाइप ही करना पड़ेगा।
अपना नाम दे दे पहले, आपके क्वेश्चन
तो तुम लोग क्वेश्चन सुनना और टाइप करना।
लाइट है और अभी तक लाइट ऑन करना है क्या? आसपास ऊपर वाला
सारी लाइट ऑन करवानी चाहिए।
Congratulations, ma'am. So in the next series of award is BBIF Outstanding Award that goes to 2025, that goes to Dr. Virendra Yadav, convener and the Chief Organizing Secretary. Congratulations, sir. So now we have this year BBIF has given travel grants to 3 students. So 2 are from Nepal and 1 is from Bangalore. So I would like to call upon Miss Pramila Banjara from Nepal to come and rise to. The second is Miss Raksha Rathiwada, student of medicine Nepal. And Apurva Srinivasan, HCG Bangalore. So I would request our chief guest to kindly encourage these students. Dr. Das, please. It would be an honor for all of us.
मेरा काम खराब हो गया। ये तो बाहर वाली आवाज आ रही है।
Thank you all and congratulations.
बाहर आओ, मैं इसके नहीं आ रहा।
Thank you, ma'am. Now I request Dr. Nani Kumar, BBIF present, to address vote of thanks and also release the abstract book of the conference.
Speakers and esteemed participants, I extend a very warm welcome to the International Biobanking Conference and Asia Pacific Forum on Therapeutics and Vaccines under the theme One Biobank, Many Impacts, Enhancing Human, Animal and Agricultural Health. This is what is reflecting the Vasudhaiva Kudumbakam the oft-spoken slogan that was repeated last year when India was having the presidency of G20. So this conference brings together experts from diverse disciplines, as was highlighted earlier also, to advance biobank science and its transformative impact on healthcare, agriculture, and animal sciences. I extend my sincere gratitude to our chief guest, Professor K.P. Singh, and our esteemed guests of honor, Professor Y. Vimala, Professor Devendranath Das, Dr. Zisis. As you said, the number of names of the Professor Ranganathan, Professor Pawan Kumar, Dr. Kavita. I hope I have covered all of them. Great thanks to all of you for your presence and invaluable contributions to research and innovation. I also wish to thank our partners, Jawaharlal Nehru University, AIRF, SCMM, One Health India Organization, BBIF, Cancer Care, and Nanomolecular Society for their unwavering support in making this conference a reality. I would also like to thank especially other people also who have helped in this, namely Professor M.S. Kaur, Executive President NMS, and his team for his valuable inputs and all suggestions and support. Local Organizing Committee at JNU, Dr. Kulika Prasad, AIRF, JNU, and her team of volunteers, Dr. Gajendra P, SCIS JNU with his NCC team, Professor Sapna Ratan Shan, SCIS, Dr. Pratima Solanki, SCNS, and all of them who have worked day and night and contributed towards managing the nitty-gritty of the event. Not to forget, of course, Professor Manurada Chaudhary, Dean of Students, Interpol Administration for providing support in materializing a marathon, NCC, NSS, and sports volunteers for this event. I wish to actually discussion for that, but the spirit is there. And with this, I officially declare the conference open and invite all participants to engage, collaborate, and contribute to shaping the future of biobanking and translational research. Let us embark together on this journey of discovery and innovation. And thank you for your presence here. And I hope you have a very fruitful learning session of these 2 days. Thank you very much. Also, I would like to actually thank Professor Shanti Pandit, Vice Chancellor JNU, Professor Rajesh Kumar Pandey, Rector 1, JNU, Professor Devendranath Das, Rector 2. I had already thanked him earlier, but I'm endorsing it once again. Thank you very much.
Thank you, ma'am. So, since many dignitaries have taken out their precious, now it is the time to release the abstract. So kindly, I request all the dignitaries to kindly release it.
हाँ देखना आगे समझना इसका वो है ना उनका बुक नोटिफिकेशन
It's a compilation of hard work of many, so we should applaud a little louder. Thank you all. Since many dignitaries have taken out their precious time out of their hectic schedule for this inaugural event, so before some of them may disperse, we must capture this to make it a moment memorable. So I would request all the delegate speakers and all dear organizing committee members to join for a commemorative photograph.
थोड़ा थोड़ा
हेलो हेलो आप पीछे आ जाओ। आ रहा है अभी कपड़े जो नहीं
Okay, thank you.
अभी तो ऐसा क्या बोल रहा था यार बंदा जो है बोल रहा है कि मैं तो सिर्फ लैपटॉप लेके आया हूँ मैं लैपटॉप कनेक्ट करके दे रहा हूँ तो पीछे क्या करना है
लैपटॉप को कनेक्ट कर देगा जो नो इनपुट सिग्नल आ रहा है वही तो करना पड़ेगा
उसको बोला कोई देगा तो ना मैं क्या करूँगा कनेक्ट क्या कनेक्ट करूँगा मैं बोला कनेक्ट करो कुछ
लिंक है वही कनेक्ट कर देगा
Okay. So there is an important announcement. All those who are interested in attending the RNA lectures, they can kindly proceed to lecture hall number 1.
You and Mr.
नहीं सब जन नहीं जा रहे एक सेशन यहाँ पे है एक उधर है
एक उधर होगा सेशन
दो सेशन यहाँ पे एक सेशन
आ जाओ बोलो फोन बात
So we'll resume our session, keynote speaker session that we were having since morning. So since Professor Sanjay Singh could not immediately join, so there was some disturbance. So now he is online and we would be having his talk. And after that we'll have a tea break. So kindly be patient.
Thank you so much. Sorry. Thank you so much. आगे वो नहीं अभी नहीं कनेक्ट नहीं है उनको बोलिए एक बार आ जाओ नहीं हो रहा हाँ बीच में कुछ के लिए जो इनोग्रेशन होगा ये चला जाएगा वहाँ पे कुछ वीडियो एक लेक्चर तो है ना हाँ कनेक्ट यही से हो रहा है भेजिए लिंक्स वगैरह वो वहाँ पे मैनेज कर दीजिए आगे यहाँ पे आप व्हाट्सएप जरा ओपन करिए एक व्हाट्सएप मैं आपको दे रहा हूँ जरा आपके सिस्टम
सर प्रेजेंटेशन ओपन हो गई
हाँ हेलो कहाँ रह गए भाई इसका आज लेकिन मैं वहाँ नहीं रहूँगा लगा देना अपना देख लीजिए हम देखेंगे जैसे आज और कल मेरा यहाँ पे कॉन्फ्रेंस है हाँ हाँ टीचर वगैरह के लिए मैंने बात किया ना तो उनको कवर करने के लिए मैं अभी जेएनयू में बैठा अब आज और कल दिन का 2 दिन ढंग से लगाना जो भी करना ठीक है
बोल दिया ये भूपेंद्र जो है ये इन्होंने कुछ रिप्लाई नहीं करा है और ये आईडी नहीं है मैंने इन्हें नंबर दिया था तो उसने कोऑर्डिनेट किया होगा उससे पूछिए
मोबाइल डेटा वहाँ पे क्या करना है
वहाँ पे भी अभी इनॉग्रेशन होगा ना छोटे छोटे शॉर्ट वीडियो देख के बना
अभी हो रहा होगा क्या स्टार्ट हो गया
नहीं स्टार्ट होगा तो ये तो आएगा ना सब कुछ देख के आते हैं उसको भी चलो आके एक बार देख लेते हैं इस पे जो भी आएगा ना उसको ऐड कर दीजिएगा बस ठीक है और एक पीडीएफ हमने भेजा है आपको उसको डाउनलोड करके यहाँ पे प्रेजेंट में ऐड कर दीजिएगा प्रेजेंट करेंगे पूछेगा स्लाइड पीडीएफ पूछेगा कहाँ से लिखना योर कंप्यूटर ठीक है ज्वाइन कर
Hello. Not yet,
Sanjay
sir.
Singh is connected. He's connected now.
So you are connected, but you are not audible.
Audible now?
Sir, just a minute, sir. Sir, आवाज.
सिर्फ 2 मिनट के लिए
Sir, just 2 minutes. Okay, sir.
Okay.
ये आप कर रहे हो
मैं उनका लैपटॉप से होगा
नीचे
So I can see my slide. If I'm audible, we can start. Hello. Okay. So I'd like to thank to organizers for inviting me to give this talk at Asia Pacific Forum on RNA Therapeutics and Vaccine. And my title for the talk today is RNA Science, a New Era in Therapeutics and Vaccines. So I'll request next, you can move to the next slide. So please go to the next slide. Can you move to the next slide? Slide is not moving. I can see the first slide, but slide is not moving. I think looks like there is some system problem. Hello. Somebody has to tell that I'm audible. And okay, the first slide. Can you go for the next slide? I think slide is okay. Thank you. So, so basically today, mRNA has in last couple of years has come as a new therapeutic opportunity. And this is moving so fast that it has entered into many areas. The vaccines has already been approved, which is if you see on the right side. But a lot of effort is happening into the cell therapy area, into genome area, therapeutic antibodies area, and protein replacement. So you can see few hundreds already trial at different stages are going, and primarily a lot of things are in the phase 3 trial. So this is a new opening. None of the therapeutic technology has taken so fast in last century as what mRNA has taken. So this is very, very important upcoming area. Next one. So on this slide, the major focus which started into the mRNA, it was mRNA vaccine and primarily in the infectious side of the vaccines. So basically COVID came, and after COVID, there is RSV, there are flu vaccines are going to hit the market. So most properly, it is focused on the area of infectious disease. And if you see what are the technologies which are going for the late stage trial and so recombinant protein, most of the vaccines were based on the recombinant technology or inactivated or viral based. But if you see the nucleic acid-based vaccine, which is primarily mRNA-based vaccine, they have just catch up from a few hundred number in last 5 years itself. So this is one of the focus area for mRNA technology, which has given prominence to mRNA today. Next one. So what we are doing at Genova, so we developed the mRNA vaccine platform, and this primarily we were working pre-COVID also on the mRNA, but when COVID came, then we focused our resources towards the COVID-19 vaccine. Next one. So it is very complex technology. So you need multiple platforms. It looks like some editing problem is there on the slide, but you can read it. So you need multiple platform if you want to succeed in mRNA. So you need microbial platform where you can produce the DNA as a template. Then you need nanolipid platform where you can produce the nanolipid, which is either inside or outside. You can delivery vehicle for mRNA. And then you have to develop its mRNA platform by combining these two. And the last one is also you need mammalian platform, basically to test that how your mRNA is expressing and how it is doing. So it is not a single simple transfection, but you have to develop the whole gambit of technologies on all 4 platforms. And then you can say, okay, now my mRNA vaccine platform is ready. So we were having a couple of them, we have worked on, and where we stand today, next 2 slides, we would like to tell you. Next one. So here, Genova decided to follow the self-amplifying mRNA technology as compared to NIH, Moderna, or BioNTech non-replicating one, they were using the pseudo, for which the Nobel Prize has been given to Drew Weissman and Catalin. And then on the other side, CureVac, who were the pioneers into this technology, a German-based company, they were not using pseudo-origin bases, and they were also using non-replicating. We decided to go into self-amplifying mRNA technology, and here we added a machinery which can multiply mRNA copies many more. And if you see on the right side of the picture, if you see there are only few of the proteins are made, but when Genova self-amplifying mRNA is applied, then it continuously produces. So what is the outcome of this? The outcome is that non-replicating mRNA produce once your vaccine has been given, next 8 to 16 hours, and then the protein signal disappears in the in vivo system. But when we are giving self-amplifying mRNA, the protein signal is available up to 20-25 days. So we predicted why we decided to go as compared to non-replicating, because we believe that immunology, for a functional immune response, you need the presence of antigen for much larger period. And if you have that, it is going to ignite your B cell, and also at the end, it is going to ignite your germ cell line, and B cell maturation will happen, and memory cell also go ahead into the same process. And that was the basic reason we decided. It was an uphill task because non-replicating, which is Moderna and Pfizer, has only 4 kb mRNA. But for us, when we add replication machinery into it, it becomes around 12 kb. So pulling out the 12 kb mRNA was not easy. It was very, very difficult. Next one. Another biggest problem was that mRNA vaccine need ultra cold temperature, -20, -80. But if you see the global picture all over the world, if you see 90% of population lives in a high temperature area, including India or LMICs. So there you don't have the cold chain system to transport this vaccine and deliver this vaccine in -80 degrees centigrade. So we realized there's a need for innovation. So we went into it and we converted our mRNA whole vaccine as a lyophilized platform, by which now it is stable at 2 to 8 degrees centigrade or stable for few months at room temperature. Next one. The second we did for ease of manufacturing, both vaccines which were approved before us, they were putting their mRNA inside the nanoparticle, but we put on the surface of nanoparticles. And that has given, first thing, it has saved a lot of mRNA. Our yields are more than 95%, because when you microfluidize under high pressure the mRNA with lipid components and form a vesicle, a lot of mRNA gone into the brokerage form, and then you have to purify, and all that kind of problem comes. Here we developed a technology by which we just by mixing nanolipid and mRNA, we can get a particular ratio, and which is highly immunogenic. Next one. And then the delivery technology also we worked on. So if you see, this is a picture which this work has been done by PATH and WHO, and I was part of that when I was at NIH, that the delivery should not be intramuscular, it should be intradermal, because if you see on the right side of the picture, all the immune defense system is available below dermis. But technology was not supporting to give the intradermal vaccination. And what is the outcome of this vaccination? First thing, it is a very fast system. Your immune response comes much faster, and it's much lower dose, the lower picture comes. So this was defined, but till we came into the picture, this technology was not existing in a well form. So we worked with a collaboration with the company PharmaJet back in Colorado, and we developed this device, and finally vaccine was delivered by this. Next one. So here if we say that we brought 4 independent innovation into it to develop the mRNA-based vaccine. First was the first left quadrangle is saying the self-amplifying. The second one on the right, it's saying the ease of manufacturing by absorbing on the surface. The third was lyophilization of the vaccine, cold nanoparticle, and the fourth is the delivery. So these are the 4 components we added into our vaccine than the existing knowledge which was available before Genova entered into this platform. Next one. And here, the primary vaccine, which was, we said, Gemcovac 19 was approved, and the booster, which is Gemcovac almost was approved, and this work came in Nature Medicine last year. And I don't want to go into the data of that because of scarcity of time, but you can look and have it, and we are very proud of our work because this was published as a full paper in Nature Medicine. Next one. Now, this paper in Nature Vaccine will come in, I think, the next issue. The manuscript have been accepted. So what was the basic problem statement with the mRNA vaccine was that the dose which you are giving, it is not stable for a longer period of time because it is all humoral response. So humoral response comes and goes away. So this is our follow-up of 90 days after vaccination. This data we developed in few hundred patients, in few hundred individuals which have been vaccinated, a part of extended our clinical trial. And we termed this paper has been accepted now in Nature Vaccine, which says that cellular immune breadth of Omicron is specific in identifying monovalent mRNA vaccine booster. Next one. So here, if you see on the left side, what was the protocol the vaccine has been given. And finally, after 90 days, we have taken the samples and we wanted to go ahead and look into what is the memory B cell is saying. And our comparator arm was Chadox vaccine, which is very, very potent and very, very, very tough vaccine. But if you see our data, it's statistically significant, and it is creating the very good memory B cell response, and also further than the resting memory B cell response you can see. So this is the basic heart of this paper, which is coming in Nature Vaccine now. Next one. The most important thing was that when we tested our Gemcovac Omicron against various, at that time coming up, the new variant, we found the cross-reactivity with the new variant was much, much higher as compared to the comparator arm vaccine, which is Chadox. And this is the basic science into it, is that because it is persistent, it is for very longer time, and it matured the functional immune response. And we haven't seen that people who got the booster dose from Gemcovac Om has came back to the hospital. Next one. So I'm just here changing the gear, and a huge opportunity is looking. I think last week Nature has published a pancreatic cancer vaccine based on mRNA from a group led by Vinod Balachandran. And here at present, this is the hottest topic. Around 98 trials are going at different stages and in different kind of cancer. So how the mRNA cancer vaccine is placing up, I would like to dwell for a few minutes, and how we are placed into that. Next one. So this is one of the very important papers. People who are interested in have this review. So the four component looks like 4 signal models of T cell activation and differentiation, and looks like T cell immune response is going to be basically future of the chemotherapy or future of the therapy which is coming. So if you see on the first left quadrangle, this is, you can say, CAR T therapy. So this is very, very important part of today. Nowadays it is going, but CAR T is only effective in liquid cancer. It is not effective in solid cancer. So a lot of research is going on into it, and I'm talking in sense of solid tumor biology, where we are keen. If you see the right side of the first quadrangle, this is also very important because cancer grows very, very fast. So you need to change the microenvironment inside the tumor cells. How we can do it? So we have another strategy for it, which is asparaginase, where we reduce the asparagine level and cancer growth reduce a lot. If you come the lower triangle of the this chart, here also one of the therapy which is going on at present, the highest level, is this PD-1 based antibodies, which is Keytruda, the highest selling monoclonal antibody in the world today, and which is around sales of $26 billion. So this is one of the area people are targeting. And the last one, which is the right side lower triangle, is the functional immune response. You have a vaccination by which you can affect it. And looks like amalgam of these 4 technologies may able to handle the cancer of solid tumor. This is very, very important, and this is where we feel that space Genoa is working today. Next one. So we believe that the T cell responses induced by our SAMA-based COVID-19 vaccine, it is beacon of hope for cancer vaccinology, and why we feel it, it is in the next slide. So if you see it here, you can see a very good CD4 T cell responses are there as compared to the ChAdOx vaccine. This is another set of data. And also, if you see the T cell responses, they are also very, very high as compared to significantly higher. But the most important, if you go for the last one, which is red block, and here, if you see that we are getting the stem cell response. So memory has been gone there. When they are seeing the next antigen, they are responding very well. And that is the basically, can we induce the specific T cell response? So in the cancer, what happened that it somehow just elude your, create illusion for your own defense system, and by which tumors are formed. If you go back and you can retrain your immune system, especially the T cell based immune system, you can retrain it, it can take care by itself to the cancer cells. So this is where we feel there is a hope for us, and this is all our 5-6 year work is now moving into this direction. Next one. The another most important, the people who are expert into the functional polyfunctional T cell response, they will be very happy to see this. And this was also a part of the paper which is coming in next few weeks. So if you see this is one of the best example of the polyfunctional T cell responses. Overall, if you say either for CD4 or CD8, and if you see the whole spectra as compared to ChAdOx, this is giving a whole set of polyfunctional T cell response. And we are very positive about it. Some of the data we have from the animal models into the cancer at present are not presenting it, but this is what is getting publication. You will see into the publication. This is next one. So if we see the platform which we have developed, the self-amplifying mRNA platform for the COVID-19 vaccine, it looks like there is what is the future implication on that? On T cell responses, polyfunctional responses, especially infectious disease. And we have recently started our program in tuberculosis. So we are moving ahead. We have some very good data in the mouse model, challenge model. But you know, mouse and monkeys are same till we reach to the human. So this is one of the very important programs going on. Then this stemness, basically, we are also looking into the CAR-T therapy, that how mRNA can transform the CAR-T therapy, can be do from vein to vein within a week time, and memory for both of them. If you see on the right side, looks like there is a breadth into this, so it can be for mutant, if there is a bird flu kind of thing. So if it is changing, it can give a good response, and again the memory. So this is the basic conclusion of how we are doing into mRNA space and what are the areas we are focusing. And I think this is my last slide. Next one. So thank you very much. And I'm so sorry I was not able to come in person to support the organizers for this meeting. But thank you very much for inviting me and going through all this trouble to have my presentation. Thank you. I cannot hear you back in any way, so I'm just waiting. If any questions I can answer, otherwise I will log off. Thank you. I'm then logging off.